Good morning, everybody. Am I audible? Am I audible? Someone please respond. Is my voice audible?
Yes, ma'am.
Okay. Thank you. So, today's class is about trematodes, their classification and their general properties. And also, we will be learning about lung flukes. Okay. So, coming to the introduction, trematodes or flukes, they are also called as flukes. They are helminths that are not segmented, they are flat and leaf-like. Okay, so they belong to the phylum Platyhelminthes and class Trematoda. So this is a classification of trematodes, systemic classification. So you can see the family is Schistosomatidae. It has Schistosoma hematobium, Mansoni, Japonicum, Mekongii and Intercalatum. The next family Zygocotyledea. It has Gastrodiscoides hominis, Watsonius watsoni. Family Fasciolidea has Fasciola hepatica, Fasciola gigantica and Fasciolopsis busci. Next family Opisthorchidea. It consists of Opisthorchis felinius, Opisthorchis viverini and Clonorchis sinensis. Heterophyidea family, Heterophyes, Metagonymous Yokogavia. So family Troglotrematidae, it consists of Paragonymous Westermani, Nanophytes, Salmonella. So this is the systemic classification. Trematodes are also classified based on the habitat. So you have blood trematodes, hepatic trematodes, intestinal trematodes and lung trematodes. Blood trematodes, you have Schistosoma hematobium, which resides in vesicle venous plexus. Then you have Schistosoma mansoni and Schistosoma japonicum. They reside in the rectal venous plexus and portal venous plexus. Next is hepatic trematodes. So Fasciola hepatica, Fasciola gigantica. Okay, they reside in the liver. Clonorchis species and Opisthorchis species, they reside in the bile duct. Next is intestinal trematodes. So in small intestine, Fasciolopsis busci, Heterophysis species, Metagonymous species, Watsonius species. In the large intestine, you have Gastrodiscoides species. So lung trematodes, you have Paragonimus westermani. Coming to the general characteristics, trematodes, they exist in three morphological forms: adult worm, egg, and larval stage. Adult worm. Morphology of adult worm. It is unsegmented. It is flattened dorso-ventrally, but some have thick fleshy bodies like cystosomes. Then their size is less than 1 mm to up to 60 mm. Okay, so they have two suckers. They are attached to the host with the help of two suckers. One is the oral sucker that is present at the anterior end, and the other is the ventral sucker which is present in the middle of the body, ventrally. So digestive system of trematodes is incomplete. Mouth is present at the anterior end, but anus is absent. Most of the trematodes are hermaphrodites. That is, they are monoecious. That means the male and the female reproductive organs are present in the same adult worm. There is exception, that is cystosomes are exception, which are dioecious. Here in cystosomes, sexes are separate. So coming to the egg. So trematodes are oviparous. That is, they lay eggs which will develop into larvae later in the environment. The eggs are operculated. That is, they have a lid-like structure. Except cystosomes, where eggs are non-operculated, but the cystosomal eggs possess a spine. So about the larvae, 5 larval forms are observed in the trematodes. They are Miracidium, Sporocyst, Pedia, in that you have first and second generation, Cercaria and Metacercaria. These are the 5 larval forms. Cystosomes, they differ from other trematodes in that they do not have Pedia and Metacercaria larval stages. They possess 2 generations of sporocyst larva. In others, there is 1 generation of sporocyst larva, but in cystosomes, they possess 2 generations of sporocyst larva. Coming to the life cycle. So trematodes, they complete their life cycle in 3 different hosts. One definitive host, that is man, and 2 intermediate hosts. So the first intermediate host is a freshwater snail or a mollusk. The second intermediate host is an aquatic plant or fish or crab. But there is exception to this also. Cystosomes do not need a second intermediate host. So mode of transmission of infection is by eating aquatic plants, fishes or crabs which have the infective form, that is Metacercaria larva. In cystosomes, Cercaria larva is the infective form and mode of transmission is by penetration of the free living Cercaria larva. So coming to the development in the definitive host, that is man. The young trematodes, they will migrate to their respective habitats, and in their habitat they will grow into adult worms, start laying eggs which are excreted in feces or in urine or in sputum depending upon the species. So after this they gain access to water. So you have three types of development of egg. First is the embryonated eggs. When laid, they will hatch to release the miracidia. This miracidia in turn will infect the intermediate host, that is snail, as in cystosomes. Next type is when laid, the eggs are unembryonated. They will first mature in water and then they will hatch to release the miracidia, which will infect suitable intermediate host. So examples for such a type is Paragonimus, Fasciola, Fasciolopsis. Then some embryonated eggs, they hatch only on ingestion by suitable snail. So examples Clonorchis, Opisthorchis and Pathogenesis. So development in the first intermediate host. So miracidium is a larval stage, which is free living larval stage. It penetrates the intermediate host like snails and mollusks. Then it transforms to sporocyst. Sporocyst become redia. This redia multiply to form second generation redia, or they will transfer to the Cercaria larval stage. Only in cystosomes there is second generation sporocyst and no radial stage. In cystosomes, Cercaria is an infective form. In the other trematodes, Metacercaria larval stage is the infective form. So in the second intermediate host, after ingestion by the fish or crab, the Cercaria will develop into Metacercarial larval stage. So about the treatment, praziquantel is a drug of choice for all the trematodal infections except fasciola, where triclobenzazole is recommended. So this is a table showing differences between cystosomes and other trematodes. So the definitive host in other trematodes: man, intermediate host, first intermediate host, snail, second is plant or fish. In cystosomes, the definitive host is man, intermediate host is snail. There is no second intermediate host. Infective form is Metacercaria larva, which is present in the second intermediate host. But in cystosomes, the infective form is Cercaria larva, which is present free in the water. Mode of transmission in other trematodes is by ingestion of the second intermediate host, that is plant, fish or crab. But in cystosomes, it is through skin penetration. The transmission of infection is through skin penetration. Eggs are operculated and there is no spine generally in the trematodes, whereas in cystosomes the eggs are non-operculated and a spine is there. So larval stages, 5 stages: Miracidium, Sporocyst, Redia, Cercaria and Metacercaria. In cystosomes, the same thing, but there is no Metacercaria, no Redia, and Sporocysts are present in 2 generations. So adult worms are hermaphrodites, that is male and female organs are present in the same worm, whereas in cystosomes they are dioecious, sexes are separate. Okay, so the female worm and male worm, the sexes are separate. The female worm will line the gynecophoric canal of the male worm. Fertilization is self-fertilization in other trematodes, whereas in cystosomes it is cross-fertilization. So that was about the general characteristics of trematodes. Now coming to lung fluke, that is Paragonimus westermanni. So the habitat of Paragonimus westermanni is the lung. So it is also known as oriental lung fluke. It causes endemic hemoptysis in man. So Paragoniasis is endemic in northeast states of India. So many cases are reported from Manipur with a prevalence of 6.7%. So as I told you, the habitat of the adult worm is in the parenchyma of lung. Morphology: adult worm is thick, fleshy, reddish brown in color, 16 mm in length and 8 mm in breadth. It is oval and the anterior end is slightly broader. Tegument is covered by spines all over. So oral sucker is present anteriorly. The ventral sucker is in the middle of the body. The excretory bladder is very large and divides the body into two equal halves. So it is a hermaphrodite with both testes and ovary present in the same worm. Eggs are oval, operculated, golden brown in color. They are unembryonated when laid. They will first mature and then continue their life cycle. So the larva, metacercarial larval stage is the infective form. It is present in flesh of second intermediate host, crab or greyfish. So other larval stages, Cercaria, Redia, Sporocyst and Miracidium. So this is a image showing morphology of the adult worm. You can see it is oval in shape. Tegument is covered all over by spines. Oral sucker is present at the anterior end. Ventral sucker is in the middle of the body. And you can see ovary and also testis. So the excretory bladder is large and it is dividing the worm into two equal halves. The last image is Kalman staining of the worm. So you can appreciate the parts. Coming to the life cycle, the host, definitive host, man, dogs or cats, two intermediate hosts are present. First intermediate host is snail. There can be genus Melania or Semisalcospira and Grotia species. Any of the genus of the snail, any one of these can be the first intermediate host. The second intermediate host are crabs or crayfishes. So mode of transmission, one acquires infection by eating uncooked or partially cooked, salted or pickled crab or crayfish. So coming to the life cycle. So it takes place in man and in the water. So man will get infected by eating uncooked crab or crayfish containing the metacercaria. So in the small intestine of man, the larva will escape and penetrate the intestinal wall and reach the peritoneal cavity. Then it will pierce the diaphragm and reach the lung. In the lungs, the larval stage will mature into adult worms. Then self-fertilization occurs. They lay unembryonated eggs. This egg is present in the sputum. It is swallowed and the egg is excreted in the feces. Then it reaches the water. There the egg develops into miracidium. This miracidium larval stage, it infects the snail, that is Melania species. So after that, the miracidium develops into sporocyst. The sporocyst converts itself into redia. Multiplication occurs and second generation redia is also formed. So you have first and second generation redia. Okay, so after that second generation redia will develop into cercarial larval stage, which has short stumpy tail. Then this cercarial stage is ingested by the crab. Then in the crab, it gets encysted and forms the metacercarial larval stage. Man will acquire infection when he eats the uncooked crab having the metacercarial larval stage, and the cycle continues. So pathogenesis. Paragonymous testimony causes pulmonary paragonymiasis and extrapulmonary paragonymiasis. Extrapulmonary type, you have cerebral paragonymiasis, cutaneous paragonymiasis, and larva migrans. So coming to the clinical features, metacercarial larval stage, it penetrates the intestinal wall of man and it migrates to the abdominal cavity. So there it can cause abdominal tenderness, nausea, vomiting. Then it migrates to the lungs, develops to adult worms and causes pulmonary paragonymiasis. There the symptoms will be productive cough with blood tinged rusty sputum with offensive fishy odor. So cerebral paragonymiasis is the most severe form. It will present as a space occupying lesion. The symptoms will be fever, headache, vomiting, motor weakness, epilepsy. Then in the cutaneous paragonymiasis, you see migratory subcutaneous nodules. So coming to the laboratory diagnosis, Paragonymous testimony, you do sputum microscopy which detects the operculated eggs. Serological tests for antibody detection, ELISA, Western blot can be done. Antigen detection by dot ELISA can be done. Imaging methods, MRI, CT scan, X-ray can detect the lesions in lungs and other organs. Then eosinophilia is observed in the peripheral blood. So coming to the treatment, praziquantel 25 mg per kg per dose, 3 doses per day for 2 days is the standard protocol. So bithionol, niclopholan, they can also be used. Triclobendazole is also recommended. Then surgical management will be needed for pulmonary or cerebral lesions. So this completes the class, that is a lung fluke, Paragonymous westermanni, and general characters of trematodes. So I'll be downloading the attendance and then I'll end the call. So please wait for that. So I'll be ending the call. Thank you all.